Today I'm going to be discussing iron and its regulation by the hormone hepcidin. I will also be discussing how hepcidin itself is regulated and its role in disease. Just for a brief overview of iron, so iron is a metal whose main function in the body is part of the heme group in the hemoglobin protein molecule. Now its function in hemoglobin is to assist with the binding and transport of oxygen. It is absorbed in the duodenum by absorptive enterocytes, and there are a number of proteins that assist with its absorption, transport, and storage. One such protein is transferrin, which facilitates the transport of iron in the bloodstream. And transferrin is an iron-binding glycoprotein. Now, iron is mostly present in the bone marrow and red cells as hemoglobin, but is also stored in large quantities in hepatocytes and macrophages as ferritin, an intracellular protein. Iron and ferritin levels are regulated by a hormone called hepcidin, which acts on the ferroportin protein. Now, ferroportin is a transmembrane transporter, which allows intracellular iron to leave a cell and enter the bloodstream. Now, iron may be absorbed into cells by a variety of mechanisms, depending on the form of iron and the type of cell. When serum iron levels are low, ferritin can be converted into iron inside the cell and be transported into the bloodstream across the cell membrane by ferroportin. However, when increased serum iron levels are present, there results in the release of hepcidin from the liver. Now, hepcidin binds to the ferroportin protein, resulting in its internalization into the cell and destruction by lysosomes. Now, without ferroportin, the ferritin in the cell can't be exported out of the cell, and therefore the increased iron basically accumulates in the cell and is absorbed, again by various mechanisms, and the serum iron levels return to normal, and the serum ferritin levels increase. With no more cellular exportation of ferritin, but continual absorption of iron into the cell, the serum iron will return to normal levels. In 2004, Nemeth et al. Were the first to link ferroportin with hepcidin. In these immunofluorescence slides, the ferroportin is stained in green, and a lysosomal marker is stained in red. They found the addition of hepcidin to ferroportin-expressing cells for 4 hours led to the degradation of ferroportin and significantly increased lysosomal markers. The study later determined that as more hepcidin was exposed to the cells, the ferritin levels in the cells increased. Hence, the iron homeostatic loop was first suggested, as increased iron levels leads to increased expression of hepcidin, and increased hepcidin leads to decreased availability of iron. Now, it's important to remember this is a normal physiological response in the body in response to iron overload. However, it is possible for too much iron to be imported into the cells, resulting in too little iron outside the cells. This is why hepcidin itself requires regulation. Now, there are many factors that have been shown to either promote or inhibit the action of hepcidin. The most important promoter is iron overload, which I've already discussed. Another important promoter is inflammation, which acts through several mediators, for example, interleukin 6 or activin B. Some inhibitors include erythropoietin, which may act through a recently discovered hormone known as erythroferrone. Hypoxia is also thought to play a role in hepcidin inhibition, possibly by increased erythropoiesis or by hypoxia-inducible factors. Now, TMP-RSS6 is a gene that codes for the metryptase 2 protein, a protein that inhibits the same iron overload pathway for hepcidin transcription, and that increased expression of this protein would result in decreased hepcidin or inhibiting the action of hepcidin. Iron overload induces the expression of bone morphogenetic protein 6, a cytokine also involved with bone and cartilage growth. Binding of BMP6 to a paired BMP receptor in hepatocytes results in the phosphorylation of receptor-associated SMAD1, 5, and 8 proteins, which, in association with the common mediator SMAD4, translocate to the nucleus, to the promoter region of the hepcidin gene, and increase the transcription of hepcidin. Now, this is known as the BMP-SMAD signaling pathway, and it is involved with many different proteins and many different genes and has many different triggers. In this case, in hepatocytes, iron overload stimulates the BMP-SMAD pathway to increase hepcidin transcription. Now, in 2008, Kautz et al. First discovered the role of the BMP6 SMAD pathway in hepcidin transcription by controlling the iron content in the diet of mice. This is iron deficient, iron balanced, iron excess. They found, like many studies previously, mice fed a diet containing excess iron had increased expression of hepcidin, as seen here. They also found, however, that the increased iron resulted in increased BMP6 expression as well as increased phosphorylated SMAD proteins. This resulted in their postulation that the BMP-SMAD pathway was directly involved with hepcidin transcription in iron. Moving on to interleukin-6, another factor that promotes hepcidin release. Inflammation triggers the release of interleukin-6, or IL-6, which binds to the IL-6 receptor present on many cells, including hepatocytes. The receptor, in association with JAK2, phosphorylates the STAT3 protein, STAT meaning signal transducers and transcription proteins. STAT3 regulates the transcription of many target genes during inflammation. For example, in hepatocytes, it upregulates the transcription of hepcidin. In 2006, Wright et al. Found that interleukin-6 is necessary for the synthesis of hepcidin during inflammation. They found that in the presence of IL-6, relative hepcidin expression was significantly increased in hepatocytes. They also found that active STAT3 was increased in the presence of IL-6. Furthermore, the study suggested that hepcidin and IL-6 were directly related in the pathogenesis of anemia of chronic disease. Erythropoietin is thought to be a key inhibitor of hepcidin. Under hypoxic conditions, erythropoietin, or EPO, is secreted from the kidneys. It targets erythroblast proliferation into red blood cells by the EPO receptor. Many studies have shown previously that erythropoietin somehow inhibits hepcidin secretion by the liver, and in 2014, Kautz et al. Discovered a new hormone known as erythroferrone, which is secreted by erythroblasts in response to erythropoietin. They found that hepcidin mRNA and serum hepcidin levels were significantly decreased in mice who were injected with recombinant erythroferrone, in contrast to mice who only received saline. They concluded erythroferrone acts directly on the liver to suppress hepcidin secretion, but the actual mechanism of erythroferrone's activity is yet unknown. Now, many diseases have been associated with hepcidin and its regulation. For example, most notably anemia of chronic disease, it has been postulated that prolonged IL-6 release results in excess hepcidin secretion, leading to the anemia seen with chronic infection, chronic malignancies, or inflammatory diseases, aka the anemia of chronic disease. Now, iron refractory iron deficiency anemia is a genetic disease due to a mutation in the matriptase 2 protein, an inhibitor of the BMP-SMAD pathway. Without this key regulator, hepcidin is secreted in excess during childhood, leading to anemia. Hereditary hemochromatosis, on the other hand, results in decreased hepcidin. It's another genetic disease of the BMP-SMAD pathway, but this time it's of the HFE gene, or sometimes the hepcidin gene itself. It results in suppressed hepcidin secretion, therefore leading to a toxic iron overload in the body. Hepcidin is also implicated in beta thalassemia, where the high erythropoietic activity is thought to suppress hepcidin secretion, hence leading to iron overload. Establishment of hepcidin as having a potential role in many diseases has led to an effort to develop a therapeutic agent, either against hepcidin secretion or promoting it. In 2010, Sato et al. Generated knock-in mice with excess human hepcidin production, leading to anemia. In these graphs, you can see that the administration of the antibody resulted in decreased intracellular iron retention and hence increased serum iron levels. Now, this may have applications in managing the anemia of chronic disease or any other disease with overexpressed hepcidin. To summarize, today I've discussed the basics of iron homeostasis and the function of hepcidin as a key regulator of iron by destroying the ferroportin protein. I've also discussed how hepcidin is regulated, most notably by iron overload, which stimulates the BMP-SMAD signaling pathway to increase hepcidin transcription. IL-6 also increases hepcidin transcription by the STAT3 pathway. And hypoxia and hence erythropoietin, on the other hand, they decrease hepcidin transcription, possibly by this new hormone known as erythropoietin. Finally, hepcidin is crucial in the development of many diseases, most notably anemia of chronic disease, and therapeutic targets involving hepcidin are currently being considered to manage these diseases. Thank you very much. This is just my references, and the primary papers are in bold.